Hello, everyone.
Yes.
Hello, everyone. Let's get started. Thank you, everyone, for joining today's seminar. Today, we are very delighted to have Dr. Jing Ma presenting a seminar today. Jing told me the last time she presented, it was 7 years ago. So now it's,
That was in the Health and Sciences building, the old one.
yeah, so now it's
No.
yeah. So brief introduction. Jing is an associate professor of biostatistics at the Fred Hutch Cancer Center within the Public Health Sciences Division, and she's also affiliate faculty in our department. She's broadly interested in network biology and high-dimensional data analysis motivated by important problems in genomics and neuroscience. And Jing received her bachelor's degree in mathematics from Fudan University and PhD in statistics from University of Michigan. And before coming to Fred Hutch, she was a postdoc at University of Pennsylvania. Welcome, Jing. Looking forward to the seminar.
Thank you, Ping. Thanks for the invitation and the introduction. I am delighted to be back in this new building. The health sciences building has always been confusing, so I'm glad I don't have to go back there. So today I want to talk about our ongoing work on integrating multi-omics data. I titled it as with applications to microbiome data analysis, but really some of the methodology I think could be useful for other omics data as well. Here's the outline. I would like, if time allows, I would like to talk about both parts. The first part is on network-based multiple testing, and the second part is on mediation analysis. Now, before I talk about the specifics of the methods, let me give you some background on microbiome multi-omics. So, microbiome research has been around for like a long time, but really it became active about 20 years ago. And then about 10 years ago, people started talking about, okay, we need to do multi-omics to really understand what microbiomes is doing and their functions. So by that, people really mean we need to sequence not only their DNA, but also their RNA and the protein, et cetera, and their metabolites, et cetera, so that we get a full picture of the functional activity of the microbial community. And then we can link their function with the immune system and host health conditions, et cetera. Despite this idea, this concept, being mentioned over nearly 10 years ago, the availability of in a population cohort, the availability of microbiome multi-omic data are still fairly limited. Especially there are very limited data on large population cohort metatranscriptomics and metaproteomics because microbial RNA are very unstable. I mean, it's a combination of biological, technological, and logistic reasons. Microbial RNA are unstable, and the cost of sequencing them is high, which really limited their scalability. On the other hand, there are a lot of metagenomics and metabolomics data because getting metabolites, they are relatively more stable. So and metabolomics have also been studied for other type of research, not just in microbiome. Most metabolomic data are from plasma samples as opposed to fecal samples. So what does that mean? What that means is that when metabolomic data are obtained from plasma samples, they usually consist of both host and microbial-derived metabolites. So it's a combination of signals. When it's from the fecal samples, we might expect to see more microbial metabolites. Okay, question.
Question.
Yeah.
So how do you distinguish the metabolites, so let's say even in plasma, from the host metabolites? So what's I mean, I could see with the metagenomics and sort of the DNA, I could see how you distinguish it. But so with metabolites, I don't understand how you distinguish it.
Okay, so the question is, how do people distinguish host and microbial metabolites? My answer to that question is I don't really know how. I think they're just linked together, tightly linked together. And so far, our understanding of microbial metabolites is very limited. And we know a few metabolites are derived from microbiome, like the butyrate. But really, there may be more, and there may be and that we just don't really know. Okay.
I'm being ignorant in the room. What's a metabolite?
Metabolite is, It's just a chemical compound. So common, like think about amino acid. They have different chemical structure, and they have like the carbon structure, like a CO, like HO2 is like it has a structure. So these chemical compounds, they have different structure, different mass, and different mass charge ratio. Yeah, very biochemical. I mean, that's as much as I can explain to you. Okay. So like I said, we expect smaller shared variation between metagenomic data and plasma metabolomic data. Still, there will be shared variation because some of the metabolites are likely to be derived from the microbiome. And with microbiome and metabolomic data, we're hoping to really learn the interactions between these two domains. So here are just some examples of the biological interactions. Yeah, I'll just show them. So the gut microbiome, they can produce short chain fatty acids that are then used by the host for energy. And in fact, several with one of my collaborators who works on allo-HCT, they actually demonstrated that certain short chain fatty acids can affect the immune system to the extent that it can affect the patient's survival and also the development of GVHD, graft versus host disease. And they are experimenting whether administering short chain fatty acids can improve their response to the transplantation. And similarly, there's also bile acids produced by the host. They are metabolized by the gut microbiome, and the resulting metabolites can influence liver function and other metabolic processes. And if you, like me, also take vitamins, and the gut microbiome can also help synthesize vitamins that are beneficial for your health.
Kind of a follow-up question on Ali's question. By looking at metabolomics, are you actually trying to distinguish whether it's from the host or from the microbial, or is it more like a pathway that you have the microbial is like they can affect host metabolomics, and maybe that's kind of the mediator for like a downstream disease outcomes?
I think you're jumping ahead. So that's some of the methods that we're trying to do will answer different questions. Yeah. Okay. So for example, yes, what are the questions we can answer? Yes, we might want to know which metabolites are from the microbiome, are actually derived from the microbe, and we may also want to define some of the uncategorized microbes or metabolites. As the technology advances, we're getting deeper and deeper sequencing. There are a lot of genome dark matters that we don't really understand. So we want to do integrative analysis. And then lastly, we may also want to know, are these metabolites mediating the association between microbiome and health condition? So let's look at the first part. So in this part, my aim is to understand the association, to learn basically relationships between microbes and metabolites. Hopefully shed light on which metabolites are derived from the microbiome. So in this type of network, each node would describe a microbe or a metabolite, and each edge would represent their relationship. So now, the relationships, as I described earlier, they can be complex and bidirectional. So, and if we, depending on the type of assumptions we're willing to make, we have to, have some compromise. So here, especially if we have cross-sectional data, the type of inferences we can learn from data are limited by the assumptions we make. And here I want to make as few assumptions as possible. So I'm just going to say my target of interest is just the pairwise association network and between microbes and metabolites. And our data are the abundances of microbes and the concentrations of metabolites from the same set of individuals.
Can you define association network? What is that?
Association network. So here, association, you can define an association network, basically use any association measure. A simple association measure could be correlation. It could be mutual information that are more generic. Yeah, that's that type of.
So if I find a correlation, I put an arrow, an edge.
Yes.
An edge.
Yes, an
Yeah,
edge.
and
No
undirected,
direction, undirected. Correct. Yeah. And this can be framed as a multiple testing problem. If we have P1 microbes and P2 metabolites, then we're basically having P1 times P2 hypothesis that we are testing, okay? For when you test multiple hypotheses, especially if you have a lot of variables, a lot of hypotheses, it's good to have some form of error control or statistical significance measure. And here enters the false discovery rate. This is just one way of quantifying the statistical significance when you're testing multiple hypotheses. Suppose we have a set of the M hypothesis, and we have some decision rule that tells us some of them are significant, some of them are not. And then we can write out this table and here and you can count the number of false positives, false negatives, etc. And the FDR is just defined as the ratio of the false positives to the total number of discoveries. And oftentimes people also work with the marginal FDR, MFDR, because it's the ratio of expectations instead of expectation of ratios. When the number of hypothesis M is large, these two are basically asymptotically equivalent. Perhaps one of the most popular method for FDR control is the Benjamini-Hochberg procedure. This is from one of the most highly cited statistics papers. It's basically, it takes input of vector p-values. So for each hypothesis, you have a p-value, and then and its operation is very simple, basically by ranking the p-values, and then you find a cutoff such that your FDR is controlled. This is conceptually very similar, and they demonstrate that this method works for both like independent hypothesis tests and also some arbitrary form of hypothesis tests. Although in practice, I often notice that it can give very conservative results. So, and later on, a really important development was given by Efron's group, which studied the mixture model, basically. So instead of studying the p-values, Efron decided to model the z-scores. So for each p-value, you can assume you have a z test statistic. So we can model the z-scores as a mixture distribution, as coming from a set of a mixture of a certain null distribution and an alternative distribution. And so Efron defined the local FDR, which is basically the probability of the hypothesis being null given the data. And this has a very natural interpretation. And then their group and later on Tony Tsai's group showed that this type of method can actually gives you a higher power by modeling the distribution of the test statistics. And then later on, more work has been done for by saying like we can further improve the power by exploiting local dependency structure. This is particularly useful if you have a lot of hypotheses and when the BH procedure can have very low power. So one particular type of dependence structure that was explored is the hidden Markov dependence. And so this result was taken from Sun and Tsai's paper, and which shows that compared to the BH method, which are these circle points, the new method that's based on the hidden Markov model has a smaller FNR, uniformly smaller false negative rate, while also controlling the false discovery rate. So this is one example telling us that we can gain more power by modeling the latent structures among the hypothesis test. Now, okay.
Is that based on assumptions or where is that information coming from?
It's based on the data structure. I think in certain applications, there are local dependencies. If you think about if you're doing the GWAS, the hypothesis that there may be, if they're coming from the same chromosome, there may be local dependencies that are suitable to be modeled in this way.
That would be additional information.
Yes, that will be additional information you use. And typically it's part of the model. You just learn the model parameters from your data and then update your local FDR. Yep.
Just a follow-up question. So, if we were going to use hidden Markov model for or either metagenomics or metabolomics, what's the handle? Just for GWAS in the genome, like it's a natural sequence.
So first of all, we are not going to use hidden Markov model for metagenomic data, and also for the hidden Markov model applied to the GWAS context, I believe it's still cross-sectional data. I think it's the dependence, like basically it's what's the state, I guess the steps, how much further, how many legs do you want your dependence to be? I believe that's a parameter in the model. I think I don't remember exactly how they estimated it, but it's part of the model parameter. Yeah. So here what I want to say is that well, I've only listed a few multiple testing procedure, but many of the existing ones, they have the same kind of limitation, which is that they require vector inputs. So in the GWAS setting, you have a vector of p-values or vector z-scores, and then you pass to these methods, they spit out a list of corrected p-values. But these type of methods are not optimized for matrix valued observations. So what are some examples of matrix valued observations? Correlations. So correlations are just a very kind of very specific example. If your interest is to test correlations between pairs of variables, then you get a matrix of z-scores. And in that case, if there are certain dependence that renders the dependence into a certain kind of network type of structure, then these existing methods, which vectorizes the observations, will not be able to capture that. By network type of structures here I refer to modular structures, such like block structures, community structures, or preferential attachment hubs, etc., etc. So some of these network topological properties have not been accounted for cannot be accounted for if you use vector inputs.
So, I mean, some question that is super basic.
Sure.
If I have p variables and I know the matrix of their covariances, I have a constraint that it's positive.
Right. Yeah.
So is that also a consideration? When you say that that when you test a square matrix, you have more structure. Is that the kind of structure that I should also be worrying about?
That is a great question. Right now I don't really consider that because yeah, I guess I haven't really thought about that. I think that could be one type of structure that is, if it's a structure that's related to its spectral property, like the Engen structure, then it could potentially be incorporated just like how the community structure is.
Yeah, it will give you inequality constraint.
Yes.
So,
yeah. Cool. Thanks for raising that up.
The kind of structure that they're talking about is on the test statistics themselves, the collection of test statistics, or whatever. Right?
The collection of test statistics. Well, as well.
Data. I mean, they might imply something about the test statistics, but it's really on the test statistics.
So, correct. So you can imagine the, let's just hypothetically, there is a true network that has some structure, and then when the biological system as it evolves, it will spit out some observations, like some of these microbiome abundances, metabolite concentrations, and then you would expect, after you calculate the correlation and stuff, you would expect these correlation statistics to be somewhat related to the underlying network That's the type of connection I'm talking about.
So any
Yeah.
structure on the underlying data unit may impose some structure on the.
On the test statistic.
depending on whatever.
Mhm. So this is where we develop this latent graph model, where suppose we have a matrix valued z scores. We model again here we are because we're interested in the association between two data types. So we are using bipartite networks because so that one side of the nodes would be microbes and the other side would be metabolites. And in this bipartite graph, I have some latent block membership or latent clusters that's denoted by Z, Z1, Z2. And then given these latent block memberships, I have the two associations or the network skeleton, they follow an independent Bernoulli distribution. And then given the network and the block membership, my z-scores would follow an independent kind of a mixture distribution, which is a mixture of some null distribution and some alternative distribution. The alternative, the null is a single like uniform null, but the alternative distributions are allowed to depend on the block memberships.
So, hold on, so the clusters, are those,
They're latent as well.
no, those are latent, no, those are latent, okay, sorry.
Yeah.
And you're going to learn the.
I'm
alpha
going
one
to
and
learn,
alpha two
yes,
as
I'm
well?
going to learn, yeah. I'm going to learn all the parameters, which are the alpha 1, alpha 2, and the pi, the probability of having connections, as well as the null and alternative distribution parameters.
But
mean-
at the end of the day, do I care about the conditional distribution given the clusters, or do I care about the association? What is the ultimate?
This is,
goal?
this is what, so this is the, yeah, because we're thinking about multiple testing. We're really like in the context that we're developing a procedure for multiple testing. So we want to use the posterior probability of the hypothesis being null to control the local FDR. But this is the ideal quantity. Again, because of our network structure, these quantities are not tractable. So we used an extra level of conditioning by conditioning on the block memberships. So this gives us the structured L value. And this will be the kind of the posterior that we use in defining the local FDR.
You relate.
Can I?
This connects to what you want.
They are connected, but the relationships are kind of there is a summation. Basically, it's a sum. You just sum up Z, you get the top.
Does allow you to get back.
Yes.
So if I,
That's true.
this is what I can work with, and this is how it relates to that, and so I'm comfortable rejecting, you know.
Yeah. In theory, like so the one tricky part is that the Z1 and Z2 are latent. So if you don't know them, it's hard to say much about them. But I guess we could do for the estimated quantities, like what is the relationship? Yeah.
Sorry,
Mhm.
would you mind going back to this slide? One thing I'm trying to. So. Given Z1 and Z2. Then you also have the structure of the network, so the network structure. You also using the network structure, any assumption of the network structure.
So the networks,
It's just a block model.
it's just a block model. It's just a block model. Yeah.
No,
It's-
so I was wondering if you were making
Yeah.
any additional assumptions.
If you remove this last equation, that's basically the block model, stochastic block model. Yeah, and we're using the stochastic block model as a model, just like the hidden Markov model.
The assumption is sort of that each block either we have.
The same
All-
null or some non-null. Yeah, in each block, I will have some yes, the introducible some null and then a single alternative, and then I can then pull all of the z values together. And then obviously you can also in the more aggressively, you can say there is a single null and a single alternative. In that case, you pull all of the pull the z-values across the blocks as well.
Sorry,
Okay.
super naive question. So is a block basically, can I understand it as like a group of metabolites that belong to the same pathway, or?
Yes,
Okay,
yes.
and the Z1 and Z2, one for the
One for the microbes, yeah.
microbe,
Yeah.
one for the.
One for the metabolites. Yes,
Thanks.
yes. So essentially, you can think about this is like a network-based bicluster. Yeah. Yeah. Okay. I think you guys have already got all of this. Basically, you know, by conditioning on the block membership, we are pulling information across observations so that we can gain power. In terms of identifiability, I think in the special case of my the null and alternative being Gaussian, we can show that this model is identifiable if under the constraints that all the elements for basically the mean and variance parameters for the null and alternative distributions are distinct across the different blocks. But this is a stronger assumption. It's not necessary. Because like I said, the model is also identifiable if there is a single alternative distribution. It's just some proof. Yeah.
You know, I'm not an expert by any means in mixture models, but I think that when you don't know anything about the mixing probabilities,
Right.
there is a problem near when they both
Yeah.
collapse, like you have
Yes, that's right.
different distributions for detecting. I don't
Yeah.
know if you would take that into consideration when you say they are different. You know, the means and the variances. The means, I guess, have to be different because you really want two components to be able.
I want to be able to separate them.
Right,
Yeah.
to separate them. But are you going to consider an asymptotics where those two components could be shifting towards null when you analyze the power?
I have not.
Mhm.
But I agree with you that it would affect the power. I think I mean empirically I do notice that if the separation is small, the power is affected.
Those mixtures are very difficult
Yeah.
to detect when the mixing probabilities
Right.
are very small.
Yes.
Or, you know, when there's almost no mixture.
Yeah. So the separation, yeah, that's the signal is small. Yeah. Yes, great point. The last part of this model would be just a parameter estimation. Well, actually, second to last part. The last part would be model selection. So parameter estimation. Here we have some latent variables, and we just use the YAM algorithm to estimate the parameters, which says that instead of working with the observed data likelihood that are difficult, we work with the complete data likelihood with respect to some distribution Q. And in theory, Q is taken as this conditional distribution of the latent variables given the data and the estimate from the previous step of your algorithm. And again, because of our network model, even that full conditional distribution is intractable. We have to use this mean field approximation to break it down to be the product of some tractable distributions. And then we update. Once we have the Q, we can maximize the evidence lower bound to get the parameters. And you just iterate until convergence. All right, so this is the last thing. The last thing is about the number of blocks. So far, up to now, the algorithm works well if you tell it how many blocks you wanted to estimate, but we still need to kind of a procedure to determine what's the optimal number of blocks. And in the context of network models, this can be done using the so-called integrated classification likelihood, which is basically a criterion that's similar to BIC. You know, it's BIC modified for network data. You just use an extra term that accounts for the performance in cluster. Before I show you some empirical results, I just want to quickly mention why we prefer this bipartite SPM, and I think you probably know this already. Well, the first reason is computational. So, so any bipartite network can also be inferred by the unipartite SBM. You basically combine, if you have a bipartite, you can combine the network to be a bigger network so that you can infer the entire network and then only take out the top right block to be your bipartite network. But as you can imagine, if you make it a bigger network and apply unipartite SBM, that's a much bigger problem, and which is computationally gonna take more time.
Sorry, when you say
Unipartite,
meaning that I
meaning
would-
the rows and the columns are the same.
Right, so I would like do a correlation also between two
Microbes.
microbiome.
Yes,
I-
yes,
I
yes.
see.
So there will be some extra, yes, there's some extra information you're learning. But if all you care about is the association between microbes and metabolites, you don't need to go through that stuff. Yeah. Yes, that's exactly the second reason. So here are some quick simulations to just to show you how it works in practice. And there are 3 different network topologies we considered. The first one is a very simple modular structure, a network with 3 biclusters, very clearly separated. And then the second is called a nested bipartite graph. In ecology, this could mean that there are some generalists that just interact with everyone. And then there are some specialists that only interact with one other organism. And the last one is a preferential bipartite graph from a preferential attachment model, which basically says the kind of the species that have more neighbors will get more neighbors. Just some basic setup. We compared the average performance over 100 replicates. One thing I do want to note is that I have to implement the Sun and Tsai procedure, assuming the null density is known. This is very important because we found that if you don't tell us the null density, its FDR is not controlled. It's inflated. Yeah. I think there are also works that says that it kind of the empirical null sometimes works better than the theoretical null, but how to correctly estimate the empirical null is challenging. So here is the plot of the FDR over the TDR, in which you can think of as the ROC curve of a few different methods as a function of the nominal significance level. So each dashed line indicates one significance level. Yeah, and so across the 3 different settings, you will notice that the proposed method has a higher ROC curve. And in setting A, its FDR is controlled. In setting B, it's actually slightly conservative. But in setting C, it's slightly anti-conservative. I think it's because in setting B and C, the network model, I just fixed the network model. I did not really generate it from a bipartite network. So there is some network model misspecification. And then we can also assess whether the clusters that we identify are correct and whether they're meaningful. In setting A, it kind of estimated the 3 biclusters mostly correctly. In setting B, it estimated the 2 biclusters in most of the time. And these 2 biclusters are separated by the degree, basically, the degree distribution of the nodes. So one that has high degree and then the rest that has low degree. That's how our method would group them. And that's in line with the underlying structure of the stochastic block model as well. And similarly, in the third setting, it also tries to group the nodes based on their degree distribution, basically. Yeah.
What you mentioned about sort of concept,
Yeah.
How, I mean, the method that you propose does not have that information. It doesn't need to know that.
It needs to. So I think it depends. It can allow, you can tell it the full null distribution, or you can tell it the mean. What I find that if you don't tell it anything at all, it tends to I don't feel like maybe there's some constraint that I have not incorporated, but I may not know which one is not, which one is alternative.
It's labeled
Yes, yes. So I need to tell it.
custom.
Yes, yes. Mhm.
Sense, but sort of, I mean, how would this, I guess, sort of with a normal distribution simulation makes sense, because sort of how would it be in the case of microbial sort of abundances and happening abundances that's
Right.
normal distribution exact.
Yeah. Well, you're really thinking ahead. I think in the context of this application, I actually did something really simple because I used some statistics that require very weak conditions to converge to a normal distribution. I did not really worry about the distribution of the statistics. But I think yeah, I think perhaps other statistics and then other distributions can be developed to allow more complex data structures. Yeah. Well, in this example, I think in this example, we just have two types of data and 49 bacteria genera and 128 metabolites. And we want to know we have two groups, normal and diseased individuals. So we want to know what are the differences, like what are the changes in the microbial metabolite associations during this disease condition. And this is how we define the test statistics. And then we used the two sample version so that we can do the two sample inference. And if you look at the distribution of the test statistic, it looks like normal, but it's not standard normal. So, and it turns out like the estimated marginal distribution from our method actually fits the distribution pretty well. This is the plot of the percent of rejected edges as a function of the significance level. In all, at all levels, I think the new method just rejects a lot more edges. Here is an illustration, a visualization of the network or the bipartite network, also clustered, also with rows and columns ordered according to the estimated clusters. And so on the left, that's just basically the heat map of the test statistics that you saw the histogram of before. And the right is the inferred graph at a very stringent FDR. And I think intuitively you can think about these blocks as microbial metabolic pathways. So some group of microbes would correlate together with groups of metabolites, and then they together affect biological function. And yeah, there are also some interpretations here. But I'm gonna summarize. Yeah. Yeah. Sure.
This is really cool. I mean, especially the results. That's a question that was pretty interesting.
Yeah.
If I were to do naively sort of two separate things.
Okay.
First doing like the biclustering of microbes and metabolites based on the logarithm of
Right.
some normalized version of that.
Mm-hmm.
And then applying the method based on the learned sort of
clusters. Right.
How would that compare? Because so you hear, the reason I'm asking and sort of I'm intrigued by this is that sort of you here sort of don't have just the clusters, but you have like holes in the clusters.
Yeah.
In a sense that sort of how would that naive approach compare to sort of your approach?
A great question. First of all, I think I did use, for example, naive biclustering as an initialization of my algorithm. So let's just say in the case of that, let's say that's a perfect estimate of the latent clustering. Then I would think you may be able to get something quite similar. Yeah.
Do you know how different the clusterings that you learn from your method are from the clusterings that you
I have not checked, but I think, yeah, I will. Yeah. Mm-hmm.
Thank you.
Yeah, sure.
Sorry, just a very quick question.
Mhm.
Did you need to adjust weighting covariates such as batch impact in either pre-processing step or in copying models?
For this one, no. For this one, I assume that the data have already been kind of normalized so that everything is comparable. Yeah, but pre-processing is always important. I just want to give a few caveats. So there are still some unresolved questions that I have, and maybe some of you have ideas. One of them is that right now I use as you know the variational EM. It can be quite slow, especially you know how can I make it scalable to larger number of variables, and I think we need some alternative model selection or model estimation procedure. And then the other thing with networks, you know, at least in the context of stochastic block model, it doesn't really work well for networks with heterogeneous degree distribution. So although to be honest, I have not explored how that would affect the performance in terms of hypothesis testing, but that may be one thing to think about as well. And then also back to Ali's question about the data statistics, and right now I'm using very naive test statistics without worrying about the distribution of the microbiome data, but perhaps that's also something we should think about. Okay, great. So I think I have maybe 15, 17 minutes or so. Hopefully I'll at least give you a flavor of what I'm trying to do and learn from you guys. So in this second part, I want to, you know, I want to study kind of mediation analysis, where in this particular problem, we have, you know, bioassays, we have microbiome data, and we also have outcome on insulin resistance. And the hypothesis, you know, based on the literature review, we know that bioassays dysregulation are linked with insulin resistance. And we also know that the microbiome has a role in converting primary bile acids into, or in other words, basically the microbiome can modify bile acids. So our question is, what is the effect of the microbiome on insulin resistance? And to what extent is this effect mediated by the bile acids? And
Why is that important?
why is that important? Well, I think so for the purpose of designing, for the purpose of designing, let's say a treatment that's based on your microbiome, you want to be able to make it like kind of know how it works. Does it target the bile acids, or do I need to develop like a bile acids based therapy? Like if a capsule that can be developed in terms of bile acids, maybe I don't need to just develop the microbiome. No. I think, you know, the concern with microbiome sometimes is volatile. It's really hard to know what's the dosage, and it's a live consortium. How to make it, package it into a treatment, a usable treatment, quantifiable treatment, is still a hard question. On the other hand, I think bile acids, they're more stable, and so maybe we could think about the treatment in that direction as well. And this is the part where I will talk about again, say microbiome that are high dimensional, they're compositional. So they're multivariate basically. How do you deal with mediation analysis with multivariate exposures? I don't know if any of you has worked on it, but my literature review tells me that it seems most of the existing causal mediation analysis are with respect to a single exposure. And then when you have multivariate exposures, they typically first do some kind of a dimensionality reduction by converting the exposures into basically independent variables, and then that allows you to study independent mediation pathways. Without doing that step, you may need to be worried about the dependence among the exposure variables. Is that a concern, Angela?
I'm not knowledgeable of the literature.
Okay.
Multiple exposures. Maybe Jing here we have another expert. I don't know much about that literature, but I don't see why dependence is the right word. I mean, I'm thinking of you know, structural dependence, not correlational dependence.
Right, right.
Like not
Directed,
association,
yeah.
but causation.
Causation,
With
yes,
the
yes, yes. Well, yes. If you have multiple exposures, obviously, if you know the causal relationship among the exposures, then the pathway is easy to build. But oftentimes you don't know. You don't know that.
I'm thinking, even if I have, like, let's say I have 2 exposures.
Mhm.
Basically, what I want to know is if there is from each one of them a direct effect into the outcome.
Mm,
Because I mean, I'm thinking of the graph. I mean, as soon as one has a direct effect, I'm done. I mean, I know that I have I cannot control the direct effect because if you're thinking of, I mean, I don't know exactly how you want to quantify the exposure itself.
right.
But
Yeah.
if the exposure is really multivariate, well, and you want to know whether you can intervene, that's why I was asking you, in the bias, whatever. Then I would say, well, I need to know if each single one will have a direct effect. Why do I care about
Right.
dependence correlation?
Yes, so that's one way of thinking about it. So in this context, we are proposing is that instead of working with individual taxon, I think we could think about them as consortium. We could design treatment in the form of a consortium. Suppose so I'm gonna tell you the story. Basically, I'm going to summarize my microbiome, the multivariate microbiome, using a scalar variable. And this scalar variable is summarized using this concept called balances. Balances, they're just a special type of log ratio. They're the ratio between 2 groups of taxa. And if indeed it's this balance that's causing changes to my outcome, and then I could just design a therapy that is perfectly mimics the proportions of these taxa as defined by this balance.
But why this and not other outcome? Why this exposure? I mean, do you have a biological reason for this?
It's one way of doing, it's one way of doing modeling. I guess
No, what I'm saying
biological,
is
yeah.
maybe this transformation, for this transformation, there is no direct effect, but maybe for some other transformation, there is, because,
Possibly,
some pieces,
yeah.
like, I think of, the vector of all the exposures, what you're doing is just one scalar transformation of all of those, and then, if you go to the next one, if you really want to decompose that vector into many transformations, maybe there is another transformation that.
It's very likely that it's very likely. But I think this is just our working model. Let's just say we'll simplify it to a scalar, a single scalar, and we're not thinking about the other. Yeah. Okay.
I guess what I'm saying is that you're already assuming when you concentrate on this, is that because you are disregarding the possibility that any other independent transformation of this one. I mean, I'm thinking like, I have, if I have 2 variables, I do it, I need 2 transformations to recover, the
Yes.
So maybe this particular one you're just testing that this particular transformation doesn't have any direct effect.
I think there is an implicit assumption here, which is that, well, I guess I'm assuming that there is a sparse set of taxa that actually has non-zero mediation effect and direct effect.
Okay.
And so I'm trying to use this as a way of selecting and identifying them, as well as estimating the
So you're
mediation
asking that
effect.
any other transformation doesn't have a causal effect on the outcome altogether? There's no direct, no indirect?
Correct.
Is that what you're assuming?
Yeah,
That
yes.
this is the only-
Yes. So this would be my latent
Right.
mediation model.
And what I'm saying is that if I have like two variables, 2 exposures, I guess X is the exposure.
X
here.
is the multivariate abundances, and Z is just a latent variable. That's why I call this exposure latent.
I'm saying like if I had 2 of these, 2 X's, I need B1 and B2, to recover the whole X. Forget about the Z because I get all confused.
Yeah.
But okay, so really writing X1 and X2 is the same as writing B1 and B2. Okay, and what you're assuming is that B2 already doesn't have
Doesn't have any effect.
any effect whatsoever.
Yeah,
So
yeah, yeah,
you're
yeah.
saying I'm going to put all my eggs in the basket of the
Yeah.
B1 because
Yeah.
I'm already making an assumption. That
Yes.
Anything else doesn't have any effect on the outcome.
Yes.
By concentrating on this and declaring that there is, no direct effect when I don't find the direct effect on this.
Yes, that's a fair way of, that's a fair way of evaluating it. Yes, yes, we do have implicit assumptions that,
Okay.
I think, it's good to clarify. Yes, Marco.
In terms of data structure, when you
Mhm.
show this graph, what is the temporality? Because before you talk.
It's
about-
cross-sectional. There's no
So
time.
that's really tough, right? Because
Yeah, yeah, yeah.
it's probably
Yeah,
feedback
yeah,
mechanism going back and forth, so.
Yes,
it's-
yes,
uh,
yes, yes,
I
yes.
mean, deviation is difficult, even when you have temporal data, it's already very difficult, cross-sectionally.
Mhm. So in that sense, this is a very simplistic, a lot of assumptions. Though I just basically tease out a lot of directions that are not, yeah.
What is the outcome? And
Insulin resistance, yes. A continuous variable as well.
Right, but you're measuring both at the same time, the microbiome.
Correct,
and
correct.
the-
I measure them at the same time.
And the biomass.
And the bile acids. Yeah. Okay, so under this very simple model, I do want to mention though, so here in the balance, if we assume this simple model, in the balance here, I have a set of taxa in the numerator, I have a set of taxa in the denominator, but they're specified by the indicator vector z, but really I don't really know which one should be in the numerator, which one should be in the denominator. So this z is a latent variable that needs to be inferred from the data. And so we just basically follow this simple linear structure equation model and a lot of assumptions that I'm not going to talk about, and that you can get some sort of indirect effect and total effect. In terms of inference, we decided to do a Bayesian inference because I think just because a lot of I think it's a conceptually very clear way of estimating the parameters, and in terms of optimization, there may be other ways. I think there may be other ways that you guys know about how to deal with it. And these are just some standard priors, independent multinomial prior for z's, and these priors allow us to get the posterior distribution. Interestingly, we can integrate out for the posterior of z. We can make it so we can integrate out other coefficients so that it only depends on the data, but it still doesn't have a closed form. And we need to use MCMC with the Metropolis Hastings algorithm. I don't know if there's anyone who's an expert in this, but we developed, we try to, coming up with different proposal distributions. And it turns out that at least so far, the acceptance rate is still very low because when you have a high dimensional vector to sample from, because we have a discrete selection problem, and each variable can be in the denominator, can be in the numerator, or it's not present in the balance at all. So there are 3 different possibilities. And because of this discrete selection issue, and I think when the number of variables, the number of bacteria is large, I say 100, the acceptance rate can get to as low as 5%. I think you can always just run your MSCMC long enough so that it stabilizes. But I think in general, a better proposal is needed. Okay. I think so. This is the data from our randomized feeding study, and like I said, it uses a crossover study design, which means that the data are first measured at baseline, and then participants are randomized to 2 groups to have 2 different diets. And then after 4-week washout period, they switch their diets. So So it's the same set of participants measured as 3 time points. But in my analysis, I did not really use that time information.
You use what, the first cross, the third?
I just treat them all as independent observations.
Combine
I combine them. That's very naive, I agree. And the so we have 23 secondary bioacids. We'll have a continuous response variable, and we also have some covariates. So because I combine them, I'm going to adjust the covariates, including the diet they have and also the diet sequence, whether they took one diet first or the other diet first. I think I did another simplification, which is to reduce the dimensionality of my mediators to just one. This is not really necessary, but as a first starting point, I just want to know if if this will simplify the analysis. And the first PC explains the 23% of total variation, and this is just a visualization of the loadings. Some of the loadings with the highest weights, they are known to be associated with insulin resistance. This is the trace plots. I kind of removed I only took the ones that are accepted in my MCMC. So I removed the ones that are rejected. So in MCMC, if you have a lot of steps and then the ones that are rejected, they will be basically the same as the previous step. So instead of plotting the trace plots through the entire MCMC chain, I removed some of the steps where the values did not change at all. So I only visualized the ones that actually changed. And the thing is, they actually stabilized pretty early, even though the acceptance rate is low. There are some, well, at least in this very preliminary naive analysis, we found there are some positive direct effect and also negative indirect effect, but the total effect is still positive. Here are some of the taxa that are selected. It's the posterior inclusion probability, and I only included the ones that have a large posterior inclusion probability in either the numerator or the denominator. So for the ones that I know have very large probability to be not in the balance, I just removed it. And some of these texts are interesting, I think, but we're still trying to make sense of it. I think, so here are just some of my takeaways of this project. And I think, like I said, I think I would like to have a better proposal distribution so that my mixing time will be faster, and also it converges, it has a higher acceptance rate, etc. Perhaps the Bayesian inference is inherently like with a discrete selection of variables is an inherently difficult problem. And I think there may be ways to relax that problem into a continuous selection problem. Just as an analogy, in the sparse regression, and people have just transformed it into a spike and slab lasso type of selection. It becomes, it's a regularized, panelized continuous selection problem. So is there any way to convert, transform this discrete selection problem into some sort of spike and slab lasso? That is also worth checking. And right now I'm working with a continuous outcome, and I think the Bayesian inference works fine. But if you have a binary outcome, so Bayesian inference, in that case, you will need to do a probability regression, and posterior sampling will involve a term that depends on the sample size, basically. That what is it? Will involve a term that require inverting some matrix that is of the size of the sample size, which makes it really which adds another computational challenge. That's, you know, if you have more samples, you're gonna be more difficult to do this inference. So I think, as I said, perhaps we can consider alternative optimization strategies instead of the Bayesian posterior sampling. I think that's all. I want to thank my collaborators and my funding, and thank you for all the questions,
Thank
which
you.
I will try to think about. Yep.
Sorry, I know I asked a lot of questions, but I promise this last one.
No worries.
A bit of a general question. I work a lot on genetics, usually after we find, for example, SNP or gene associated with certain disease, well, we try to do validation studies, either an independent cohort or look at the functions of the SNP gene, try to validate that's actually a true finding. So for example, in your case, if you learn a network, there's a relationship between metabolomics and microbiome. Like, what are the next steps to validate those findings?
Well, yeah, naturally, we also need to do validation, just following a similar strategy. I think, yeah, right now the data analysis that I present, they're using public data, but we want to apply it to this data set from the study of Latinos. And I think they have microbiome and metabolomic data, but we're also trying to work with other international cohorts to get validation, so that we can, yes, validation is important. Yeah.
We exhausted ourselves.
Thank you. Oh, of course, thank you. It's been a pleasure.
Thank you, everyone. Thanks.
Mhm.